Okay, let's unpack this. We're diving into a topic that is both medically challenging and, frankly, just heartbreaking.
It really is.
We're talking about biliary atresia, or BA. It's a devastating condition in newborns where the bile ducts are either blocked or missing entirely.
And it's an obstructive cholangiopathy. I mean, globally, it is the single most common reason for liver transplantation in children. It's that serious.
For years, we've known there's a link to rotavirus RV.
Right, the same virus that, causes that awful but usually manageable diarrhea in older kids.
But in newborns, when it goes systemic, it's associated with BA.
And that connection has always been a bit of a mystery. Why does a gut virus suddenly cause lethal liver damage in a baby?
So for this deep dive, we looked at a stack of sources detailing some incredible high-tech research.
Including single-cell RNA sequencing, which is just amazing. They looked at both RV-infected neonatal mice and human infants with BA.
And our mission today is to distill what they found, because the core discovery is that the tragedy isn't just the virus.
No, it's a precise, almost self-sabotaging metabolic flaw. It's driven by the infant's own immune response. We're talking about an axis involving iron, an immune signal, and a specific hormone.
Which creates what we're calling the ironclad paradox.
Exactly. And understanding this is crucial because, the existing RV vaccines protect against the gut infection, but they don't stop this. They don't stop this systemic devastation. We need new targets.
So let's start with the hard evidence. What did the sources find that definitively links the virus to the disease?
Well, the proof of exposure was just overwhelming. When they looked at infants with BA, they had significantly increased levels of rotavirus-specific antibodies.
All of them. IgG, IgM.
All of them. IgG, IgM, and IgA. It suggests the body had mounted a really serious full-scale response to this virus.
But the real kicker was actually seeing the virus in the liver itself, right?
Absolutely. That was the smoking gun. In liver biopsies from BA patients, they detected this structural viral protein, VP6.
And it was in a shocking number of cases.
A shocking 81.5% of them. And this protein wasn't just, floating around. It was embedded deep in the tissue.
Where specifically?
In the hepatic macrophages, so the liver's main immune cleanup crew, and also in the cells lining the bile ducts. This confirms it. The virus has gone systemic and set up shop in the liver.
Okay, so this is where the paradox really starts to take shape. We have a clear invader, the RV, and the body clearly knows it's there.
Right, you see the antibodies.
So how did the immune system respond?
Well, the initial response was exactly the right one. When a cell detects a virus, the first responder is always the Type I Interferon pathway. You can call it IFN-I.
And this is like the body's big system-wide antiviral alarm.
Neighborhood watch alarm, yeah. It rings loudly, telling every single cell to lock down and prepare for an invasion. And what the researchers found was that BA patients showed enhanced IFN-I signaling.
So the alarm was blaring.
It was deafening. They saw elevated MX1 expression, which is a classic marker of this alarm, in the affected liver cells. And they saw increased IFN-beta concentration in the blood.
So every cell is on high alert. The alarm is at maximum volume. But what was the contradictory result?
Despite that powerful, enhanced antiviral signal, these same tissues were showing high levels of rotavirus replication.
So the defense was active, but completely useless.
Completely ineffective. It's like the emergency broadcast is screaming, but the virus is just waltzing in the front door and redecorating the place. This is what made the scientists realize the problem wasn't a lack of an immune response, it was a compromised one.
Okay, so the immune system is actively failing. And to find out why, they had to look beyond just the immune cells.
They had to dive into basic metabolism, using that single-cell analysis on tissue from the gut and the liver.
And that pivot led them straight to iron.
Exactly, iron. It's essential for life, of course, but in excess, it's profoundly toxic. It drives oxidative stress.
So to understand the failure in BA, you have to first understand how a healthy baby manages iron.
You do. The gut acts as this crucial gatekeeper. There are two main transporters. One, SLC11A2, lets iron in for absorption. The other, and this is the star of our story, is SLC40A1.
That's the export protein.
That is the vital export protein. And in a healthy infant, these are polarized. The whole system is designed to facilitate iron efflux, meaning it actively pushes any excess iron out of the circulation through the intestinal wall and into the stool to be thrown away.
So it's a protective flow, getting rid of the dangerous excess.
It's a critical protective mechanism to prevent iron toxicity in the liver and, well, the whole system.
So what happens when rotavirus triggers this persistent, compromised alarm state in the BA infant?
That protective flow is just catastrophically disrupted. The crucial export protein, SLC40A1, is reduced in quantity and it's found in the wrong place. Its localization is aberrant.
The gate is closed.
The gate is closed and iron gets trapped inside. This leads to a double whammy. A massive systemic iron overload, so you see high ferritin, iron precipitating in the liver, coupled with luminal iron deficiency.
Meaning very little iron in the stool.
Right. The iron can't get out. It's trapped inside the body, where it becomes a ticking time bomb.
That is a perfect transition point. So just to recap for everyone listening, we've established rotavirus is strongly linked to BA. We know BA infants have this paradox. There's a strong antiviral alarm, but the virus isn't cleared. And the metabolic fallout from this is systemic iron overload, because that key iron exporter, SLC40A1, is shut down.
And now we get to see what that trapped iron actually does.
Okay, so what is the consequence of all this trapped iron?
The consequence is the ultimate pathology. Tissue destruction via a process called ferroptosis.
Ferroptosis, it sounds intense. How is it different from, say, normal apoptosis or programmed cell suicide?
Apoptosis is clean, it's programmed, it's tidy, the cell packages itself up nicely. Ferroptosis is messy, it's violent, and it is specifically an iron-dependent form of cell death.
Driven by what?
By massive lipid peroxidation. So think of it this way. When you have too much free iron floating around under oxidative stress, it literally starts to rust the fatty membranes of the cell, the lipids.
So the cell dies from internal rust.
That's a great way to put it. It effectively rusts from the inside out.
And normally the body has a shield against this, right?
It does, a crucial one. It's an enzyme called GPX4 glutathione peroxidase 4. It uses glutathione, or GSH, to neutralize those damaging reactive oxygen species before they can touch the lipids.
But in BA, that shield is gone.
It's taken away, exactly. The enterocytes and liver cells in BA patients showed a devastating decrease in GPX4. And the evidence under the microscope was... it was harrowing.
What do they see?
Severe mitochondrial damage, so the cell's powerhouse is collapsing, plus a massive buildup of specific oxidized lipids, especially things called oxidized phosphatidylethanolamines and 4-HNE.
And seeing those specific markers is what confirms its ferroptosis.
That is a confirmation. It was the key driver of the damage in the bile duct cells leading to the blockage.
So we have the victim, the liver cell, dying by ferroptosis, and we have the weapon, the toxic iron overload. But we still need the puppet master.
Right.
What is actually shutting down that crucial iron exporter, SLC40A1?
That would be a peptide hormone called hepcidin. It's encoded by the HAMA-P gene, and hepcidin is the master regulator of iron homeostasis in the body.
And its function is simple.
Very simple. It finds the SLC40A1 exporter protein, and it causes it to be internalized and degraded. So if you have too much hepcidin, you cannot export iron, period.
Okay, so the next question is obvious. What triggered the massive sustained spike in hepcidin?
It was the persistent Type I interferon signaling, that chronic antiviral alarm we talked about.
The alarm itself is the trigger.
The alarm itself is the trigger. The sources show that IFN-alpha and IFN-beta directly upregulate the HAMP gene, the hepcidin gene, in immune cells, specifically in bone marrow-derived macrophages.
And the level shot up.
Dramatically. Hepcidin levels in the BA liver were 4.5-fold higher than controls, and this spike correlated perfectly with the loss of the SLC40A1 iron exporter.
So the immune defense signal, which was supposed to fight the virus, was unintentionally activating the systemic lockdown on iron.
Yes, and using their advanced mapping tools, the researchers could even pinpoint the specific cells responsible in the liver.
What were they?
They were TREM2 positive macrophages. The researchers dubbed them BD-Lamb's bile duct-adjacent lipid-associated macrophages.
So these are highly specialized immune cells.
Very specialized. And they were enriched right near the sites of damage, the fibrotic bile ducts. And these were the cells co-expressing the two key genes, TREM2 and HAMP, which is hepcidin.
So they're the local mediators driving this iron problem right where the damage is happening.
Precisely.
Wait a second. If the macrophages are trapping the iron, and that's causing cell death all around them, what's happening to the rotavirus that's still inside those same macrophages? This is the final ultimate irony, isn't it?
It is the ultimate act of biological self-sabotage. You'd think trapping the virus inside an iron-filled cell would kill it.
Right.
But by trapping the iron, they created an iron-rich environment. And the sources proved that this iron accumulation doesn't kill the virus. It enhances it.
The iron actually feeds the virus.
Essentially, yes. In their mouse model experiments, they specifically forced iron to accumulate in macrophages. These were the SLC40A1 MKO mice. And those mice showed exacerbated liver damage and enhanced RV replication.
Even with the high interferon alarm.
Even with their heightened IFN-I responses.
Yeah.
It turns out the iron accumulation enhances viral RNA translation through an iron-binding protein called ABCE1.
Wow, so the macrophage becomes an iron-rich factory for the virus to multiply in.
A high-capacity factory fueled by the very defense mechanism, that chronic IFN-I alarm that was meant to protect the baby.
That is a truly chilling loop. Okay, let's pause for a second. Quick summary check. The lethal damage is ferroptosis, driven by trapped iron. The core sequence is persistent IFN-I signaling triggers high hepcidin, which degrades the iron exporter SLC40A1.
Crucially, this leads to macrophage iron overload, which paradoxically lets the virus replicate, making the whole defense useless and just fueling more injury.
We've established the why and the how. Now let's get to the solutions.
Okay, so once you understand that precise pathway, IFN-I to hepcidin to iron to ferroptosis, you suddenly have a bunch of clear therapeutic targets.
And they tested them in the mouse models.
They did. They validated the whole mechanism. If they blocked IFN-I signaling, or if they inhibited an enzyme called JAK, or used iron chelation.
Like deferoxamine?
Exactly, deferoxamine. Or even just gave vitamin E, which is a powerful ferroptosis inhibitor. All of these interventions successfully reduced tissue damage and, importantly, improved viral clearance.
So the pathway is robustly confirmed. But the real breakthrough, which was hinted at by the single-cell data, wasn't some complex new drug.
No, it was a common, inexpensive, essential nutrient, folic acid, Fa.
Which is pretty surprising. Why did they even think to try that?
Well, the CRNA-seq data initially showed that BA patients had impaired folate absorption. A key transporter gene, SLC46A1, was reduced, so it suggested a metabolic deficiency. The researchers decided to test folic acid or calcium folate TF, maybe just thinking it would address that nutritional gap.
But its effect was way more powerful than just supplementation. How did it interfere with this whole vicious iron hepcidin cycle?
It turned out to be this amazing multifunctional tool. It hit every critical point in the cascade all at once.
How so?
When they treated the RV-infected mice with folic acid, it directly suppressed the upstream drivers, both IFN-beta and hepcidin.
So it calmed the immune alarm and lowered the hepcidin.
Exactly. And by doing that, it restored the function of the SLC40A1 iron exporter. It successfully reversed the systemic iron overload.
So it reopened the drain on the iron trap.
Precisely. It corrected the metabolic dysregulation, it reduced the numbers of those damaging TREM2 positive macrophages, and it improved the neonatal pathology. It hit the inflammation, the metabolic failure, and the viral persistence all at once. The potential was immediately obvious.
And this is the moment where it moves from the lab bench to the bedside. The sources cite an open-label Chinese clinical trial. What were the results? This is the ultimate so what?
The clinical results are genuinely game-changing, especially for a disease that's so reliant on difficult surgeries. They had 19 BA infants receiving a tiny dose of folic acid, just 0.4 milligrams a day for 6 months.
And they compared them against a control group.
Yes, against 38 matched controls who just got the standard care. And the first key outcome they looked at was post-operative cholangitis.
That's a severe inflammation complication after the surgery, right?
It is. It's a severe infection and inflammation that often happens after the corrective surgery, and it really compromises the outcome.
And that complication rate dropped. How dramatically?
It plummeted from 74% in the control group down to just 21% in the folic acid group at 6 months. That is a massive, massive reduction in the failure rate of the initial surgery.
But the most important metric for BA is native liver survival. Can the baby keep their own liver?
That's the bottom line. And after 2 years, the liver transplantation rates were significantly lower in the FA-treated group. They dropped from 41.1% in the untreated group to only 11.1% in the folic acid group.
That's a 73% improvement in native liver survival.
It's huge, and biochemically, it all lined up. Folic acid restored iron homeostasis, it reduced the inflammatory IFN-I and hepcidin levels, and critically, it mitigated the oxidative damage. It stopped the ferroptosis.
It's just an incredible result from a simple established nutrient.
It's a huge step forward for managing infant disease.
What a remarkable journey this has been. I mean, we started with biliary atresia, this mysterious lethal neonatal disease linked to rotavirus.
Right.
And we found the core problem isn't just the virus, it's this metabolic self-sabotage.
Yeah.
The persistent immune alarm, IFN-I, triggers the hormone hepcidin.
which then locks iron inside the macrophages, leading to this lethal cell death, ferroptosis.
And most ironically, that iron overload then helps the virus itself survive, making the immune system an unintentional accomplice in its own organ's failure.
The body's attempt to fight the infection inadvertently creates the perfect toxic, virus-friendly environment.
And the treatment, a simple, effective one. Folic acid. It targeted the entire cascade, calmed the inflammation, restored the iron export, and stopped the cell death. It really establishes this hepcidin-iron axis as central to biliary atresia.
It does. But the study leaves us with one really interesting nuance about immunity. The researchers noted that an early transient infusion of IFN-I was actually protective in the mouse model.
Oh, that's interesting.
It was only the persistent IFN-I signaling, the chronic sustained alarm seen in the BA patients, that drove the disastrous outcome. This really highlights that the duration of an immune signal is just as critical as its presence.
So the signal itself isn't bad. Its persistence in the face of this metabolic vulnerability is what proves lethal.
Exactly. It's about timing and context.
That's a powerful distinction. So for you, the listener, to consider, given this amazing success using folic acid to address the fallout of one neonatal viral infection, where else could this iron axis approach be applied?
That's the provocative question, isn't it? Could similar, simple iron-regulating therapies be effective against other complex viral-related diseases in infants that are currently so hard to treat?
A lot to think about, and certainly grounds for some exciting new research.
Without a doubt.
Absolutely. That was The Deep Dive. We'll see you next time.